Hello students, good morning to you all. Myself I am Dr. M. Indira, Associate Professor in Department of Biotechnology, Vignan's Foundation for Science, Technology and Research, Andhra Pradesh, India. Along with my colleague, Dr. K. Prakash Narayan Reddy, we both are giving a series of lectures related to the course Microbiology, designed for second year BTech Biotechnology students. So, we are in first unit where in the previous lectures we covered introduction to microbiology, discovery of microorganisms, spontaneous generation theory, germ theory of disease. So, today we are going to learn about fifth topic, that is major contributions and events in the field of microbiology. If you see, in previous classes, we observed that the microorganisms, after the discovery of Antony von Leeuwenhoek, the microbiology came to existence. We all wondered how the microorganisms were discovered by Antony von Leeuwenhoek, how the microbiology field came into existence. If you see this microbiology is, we can divide into three eras. So, the total long history of this microbiology we can divide into three eras. One is the discovery era, and second one is the transition era, and the third one is the modern era. Nothing but development era or modern era. So, several scientists, they did experiments, and this microbiology has long rich history where several scientists, they developed the hypothesis, they developed the experimentations, they challenged the theories, grudges, and finally the discoveries of each and every scientist, they contributed their inventions in this particular microbiology field. So, in this topic, we are going to learn what are the discoveries and inventions of the scientists who contributed to the microbiology field. Because being a biotechnologist, we have to learn about the microorganisms. Nowadays in the market, the biological products are produced from microorganisms. Majority of the products are microbial origin. So, the microorganisms discovery started in the year 17th century after the invention of the device, that is microscope. Later, scientists, they studied about all the microorganisms, their characteristic features, their importance in each and every field. So, we will go through all the inventions and discoveries in this topic. The objective of this topic is to review the historical events in the field of microbiology and to overview the discoveries and inventions related to the microbiology and its allied fields. So, after gone through this topic, the historical events in the microbiology field will be known and the inventions and discoveries of the scientists contributed to the microbiology field will be known. We will see what are those inventions and discoveries one by one. If you see this microbiology we can divide into three eras. So, these are the three eras. First era is the discovery era where the microorganisms were discovered by the great scientist Antony von Leeuwenhoek. Next period is the transition period where the spontaneous generation theory and germ theory, these theories were proved and disproved. That means some people proposed one theory and some people opposed a particular theory. So, opposes and proposes, so all together happen in this transition period. and golden age of microbiology. This is nothing but development and modern era of the microbiology, where the Louis Pasteur and Robert Koch, they contributed a lot to the microbiology field, particularly laboratory, where they developed technical skills in order to handle the microorganisms. So till we are following those technical skills in our laboratory for handling the microorganisms and for studying the microorganisms. So we will see each and every invention. Okay. First, Antony von Leeuwenhoek. Antony von Leeuwenhoek, actually he is a cloth merchant. He is a cloth merchant but having the hobby of making the lenses. Antony von Leeuwenhoek, he is having the hobby of making lenses. He made lenses and he increased the magnification of the lenses from 50 to 300 fold. After making these lenses, he used to observe the cloth material, the quality of the cloth material. Not only the cloth material, he used these lenses for observing the various specimens, various samples. He observed water bodies, he observed his own samples, his own blood sample, his own sperm sample. The first person who observed microorganisms, particularly living organisms, by Antony von Leeuwenhoek. Antony von Leeuwenhoek is a Dutch tradesman. Actually, he is a cloth merchant. He used to sell the cloth materials and he is having the hobby of making the lenses. So, he magnified his lenses and increased the fold, magnification fold from 50 to 300 fold. So, by increasing this magnification of lenses, he observed several specimens. He observed the quality of cloth material, he observed the own samples, own blood sample, own sperm sample and his teeth scrapings. Not only these samples, he observed the water bodies also. So, he is considered as the father of microbiology. He improved his lens, he made microscope. Using his microscope, he observed all the specimens. So, what are those specimens? He observed protists, protozoans, he observed the blood cells, he observed the sperm cells. So, like this, he observed several specimens and several samples for observation, as well as he drawn the pictures and submitted his findings to the Royal Society of London. If you see here, he observed single-celled organisms. See here, these are the various pictures observed by Antony von Leeuwenhoek. So, this is the yeast cell and this is the bacterial cell and these are the white blood cells, sperm cells. So, he has taken his own cells and he observed blood cells as well as sperm cells. And protist, bacterial organism and yeast organism was observed by Antony von Leeuwenhoek. He observed muscle fibers, bacteria, spermatozoa and blood capillaries. How the blood flow in the capillaries. So, after observation, he coined the term living organisms as animalcules. Animalcules means nothing but little animals. So, he coined the term animalcules and whatever he observed, all his findings, he drawn in the form of pictures. So, like this. And here you can observe different shapes of organisms, particularly the bacterial organisms, rod shaped, and then here spiral shaped, bunch type of the shape. This is the cocci shape and spiral form. Like this, he has written his findings in the form of letters and submitted his findings to the Royal Society of the London. So, he is the one who invented or who discovered microorganisms. Because of the discovery of these microorganisms, the microbiology field came into existence. So, this is the starting period. So, earlier in 15th century, the microscope was discovered, invented, but later The Antony von Leeuwenhoek, he modified and he built microscope and he made lenses and increased magnification of the lenses, and he is the first person who observed living organisms, particularly single-celled organisms. So, he is considered as one of the father of the microbiology. So, whatever his findings, he submitted his findings to the Royal Society of London. Later, another scientist, Robert Hooke. Robert Hooke also contributed to the microbiology field. He observed the cells and he coined the term cell. So, he observed the cork tissue. This is the cork tissue, and this is the microscope developed by Robert Hooke. So, if you observe here, so this is the eyepiece and objective lens, and here this is the specimen holder. So, this image you can see in broader view, oil lamp, using oil lamp and water flask, he kept the specimen here and using his eyepiece and objective lens, he observed the specimens. He observed this cork tissue, he observed insects, he observed the sponges, bryogens and foraminifera and bird feathers also. So, he also observed various specimens under his microscope, and whatever the findings and observations he published in the form of the book that is called Micrographia. Actually, he is a philosopher. What is the meaning of philosopher? Nothing but academician. And polymath, he is having the wide knowledge. So, architect. So, he is having the, so he found the pictures and observed all the specimens and published his findings in the form of book that is called Micrographia. So, Robert Hooke is the first person who coined the term cell. So, this cell consists of tiny compartments. It just like a honeycomb structure. Whatever the cork tissue he observed it looks like a honeycomb structure, and each compartment is similar to the other one. So, he coined each compartment as a cell. So, the term cell was described by Robert Hooke. Later, Edward Jenner. So, he is the person who saved millions of lives. Till now, we are affecting with several diseases. Every day we encountered with several diseases. So, because of his invention and because of his discovery of vaccination, we all are safe now. So, how he developed vaccination? So, we will see. Edward Jenner, he is the physician and he is the pioneer of the smallpox vaccine. So, if you see this one, smallpox, what is this smallpox? Smallpox is a disease caused by the virus, smallpox virus. So, he observed how the disease occurs because of this smallpox virus, and he discovered this smallpox vaccine. So, he is the person who saved more lives than the work of any other scientist. Okay. So, he is considered as the father of immunology. So, because of this vaccine development, now people started studying about the immune system. If any pathogen enters into the body, our immune system gets activated. What is our immune system? Our immune system nothing but consists of immune cells. What are those immune cells? Immune cells are white blood cells and lymphocytes, T and B lymphocytes. So, they encounter pathogen. If any pathogen entered into our body, encountered by these immune cells, and immune cells fight against this pathogen and removes the pathogen from our body. That is called immune response. So, the immunization and the vaccine, so the theory was proposed by this Edward Jenner. See here, this Edward Jenner, he observed how this smallpox disease is causing infection in the children. So at the same time, another lady, she is Mary Motley Montagu. Lady Mary Motley Montagu. She is the wife of the British ambassador of the Constantinople. So in that area, she observed the positive effects of variolation. Variolation is nothing but inoculation. Inoculation of a pathogen or any organism into a body of a human or host, it results in the development of immune response. So the lady, she observed the positive effect of this variolation in the native population. Okay, so in the similar way, this Edward Jenner also observed this variolation process in the children. If you see here, he observed that not only in the children, he observed in milkmaids. Who are milkmaids? The lady or the woman who collects the milk from cow or buffalo. So those people are called as milkmaids. So the milkmaid, she used to collect every day the milk from cattle. So when she is collecting, she was protected from the disease, that is the smallpox. Okay. So how she was protected from smallpox? That is the observation observed by Edward Jenner. So this milkmaid people, so every day they are collecting the milk. So when they are collecting the milk, generally the cowpox virus caused the disease in the cattle. What is that disease? Cowpox disease. So this cowpox disease was caused by the virus, that is the cowpox virus, particularly in the cows. So when they are collecting the milk, these milkmaid people exposed to that virus, and these virus, cowpox virus and smallpox virus, they belong to the same family and their genetic material is similar, but compared with smallpox, this cowpox is less virulent in nature. So when these milkmaid people exposed to this cowpox, it is less virulent. So they recovered from this cowpox disease. Not only that, they developed the antibodies in their body. So those antibodies, they fight against this smallpox. That means they protected from this smallpox virus. So this observation observed by Edward Jenner. So he postulated. What is that postulation? That is the assumption. What he assumed, these milkmaid people, every day they are collecting the milk exposed to this cowpox virus and they developed immune response. Because of the immune response exists in their bodies, so they were protected from this smallpox disease. So when they exposed to the smallpox virus, these people protected from this smallpox virus. This is the assumption hypothesis developed by this Edward Jenner. What he did, he collected the pus. What is that pus? From the blisters. What are these blisters? Those are the nothing but water, what is that called? Blisters. Those blisters formed on the skin. So he collected the pus from these blisters and he injected this pus into the 8-year-old boy. 8-year-old boy. So later this boy recovered from this smallpox vaccine. So that is the experimentation done by the Edward Jenner. So because of this invention, he was awarded with Nobel Prize. So Edward Jenner is the father of immunology. He developed the vaccination procedure and he saved millions of people. So now we are affected by a great pandemic and several scientists, they are developing the vaccines for COVID-19. So the vaccine is nothing but the compound or the microorganism it is already exist in the nature. So this microorganism, it protects. The microorganism itself causes the disease. The same microorganism, if we remove its virulent nature, the microorganism acts as a vaccine, and if this less virulent microorganism injected into a person, the person develops the immune response. So he will be or she will be protected from that particular infection. So this process was developed by the great scientist Edward Jenner. Next, another scientist, Joseph Lister. He developed antiseptic surgery. So what is this antiseptic surgery? Actually, he is a surgeon. Here in this picture you can observe. So they are performing the operation. See here, they are performing the operations. So when he is the surgeon, Joseph Lister, when he is performing the operations, he observed that after post-operation, several of these patients were infected with wounds. So this is the observation observed by Joseph Lister. What he did, he introduced this one, carbolic acid, nothing but phenol. So this carbolic acid solution is used as a sterilizing agent to avoid the contamination. Why? Because they are using this operation tools or operation devices or this surgical instruments. So these surgical instruments he sterilized with this phenol solution. So to avoid the contamination. Contamination is nothing but microorganisms. So he sterilized the surgical instruments and he performed operations. Later he found that after post-operation, the infectious rate was dropped down. So that is the observation made by the Joseph Lister, and he developed the antiseptic surgery. So like this, the antiseptic surgery process was introduced by Joseph Lister. Next, Robert Koch. So Robert Koch and Louis Pasteur, they contributed a lot to the modern biology. So if you see in this modern biology, they developed several technologies and several techniques related to microorganism, microbiology and its allied fields. Particularly this Robert Koch contributed a lot to the practical bacteriology. So he is the founder of modern bacteriology. So he developed several techniques and several technologies. We will see what are those. So he studied about these diseases, tuberculosis, cholera and anthrax. So by studying this anthrax organism and anthrax causing organism and anthrax disease, he proposed germ theory of disease. We will see, we have seen in the previous lecture, that is germ theory of disease. That means the disease is caused by causative agent, that is microorganism. So he developed the postulates. Those postulates are used for identifying the relationship between disease and causative agent. So the Koch improved the laboratory technologies, not only the study of the diseases. So the disease and disease causative agent, he developed several technologies along with his team, with students. So those techniques and technologies still we are practicing in our laboratory. Okay, so what are those techniques? So they developed this Petri plate device. They developed pure culture technique, isolation of single pure culture by serial dilution procedure. This pure culture development technique was proposed by Robert Koch and his team, and they used solidifying agent as agar instead of gelatin. So agar is having the ability to melt at high temperatures and it won't be degraded by microorganisms, and it is cooled at room temperature, so it forms the gel. So this solidifying agent is added to the broth medium, makes the solid gel, and this solid gel is used for growing the organism and isolating the microorganisms. So several key discoveries are there in the Robert Koch in his laboratory. So Robert Koch and his team contributed a lot to the practicals related to the bacteriology. Next, another great scientist who is Louis Pasteur. So Louis Pasteur, he disproved spontaneous generation theory. So before this modern era, so in the transition era, several scientists and several community people and the ancient people, they all are thought that the living organisms arise from non-living matter. So they thought that the mice comes from corn grains. It comes from sweaty underwear, and the frogs comes from the raindrops, and the flies comes from the maggots. So like this, several beliefs. So they thought that the living organisms arise from non-living matter. So several scientists involved in this spontaneous generation theory. Some people, they proposed spontaneous generation theory, and some people opposed spontaneous generation theory. That means challenges are there, experimentations are there, and acceptances are there. So good ideas are there and bad ideas are there. So scientifically they did experiments and they proved and disproved the theories. So Louis Pasteur also performed experiments and disproved the spontaneous generation theory. So not only this theory, he developed the principle of vaccination. He also studied about the diseases, anthrax disease. So he developed the vaccines for this rabies and anthrax. So not only this vaccines development, he studied about the fermentation process. What is fermentation? Fermentation is nothing but converting the organic compounds into simpler molecules by the action of microorganisms. So this microbial fermentation process was developed by, studied by Louis Pasteur. And another technique, pasteurization. So in milk, dairy industries, we are seeing this pasteurization technique, where the milk is sterilized by this pasteurization process, where the milk is exposed to high temperature and short span of time. For example, say 161 degrees Fahrenheit, time period 15 seconds. So when exposed to high temperature, the microorganisms present in the milk gets destroyed. So the microorganism free milk, sterilized milk is pasteurized by this technique. So nowadays we can observe the milk packets consist of pasteurized milk. That means the milk is treated with this pasteurization process. So he also studied about the relationship between disease and causative agent. So he proposed this germ theory of disease. And again, particularly this concept is applied in the clinical medicine, where we can identify the disease and the disease causative agent. So he is the father of germ theory. So Louis Pasteur is the father of germ theory. So not only these studies, he also studied about the wine fermentation. That means he observed that microorganisms are spoiling the wine. So for this, he developed this pasteurization technique to kill the microorganisms. And here you can observe the pasteurization, that is the HTST method. What is HTST method? Means high temperature, short time. So the temperature is 161 degrees Fahrenheit and time period is 15 seconds. So the pasteurization technique and the fermentation process and the vaccination was developed by this Louis Pasteur. So along with this Ferdinand Cohen, he contributed a lot to the microbiology field. So he is also considered as a father of microbiology. So, Robert Koch and Louis Pasteur, these two scientists are parallel and they discovered lot of inventions, lot of technologies and techniques related to the microorganisms and microbiology field. So, this is the discoveries of Louis Pasteur. Here you can observe that contributed to save millions of lives, and germ theory of disease was proposed, disproved spontaneous generation theory, law of biogenesis was proposed. What is biogenesis? Biogenesis is nothing but living organisms arise from pre-existing living organisms only, not from the non-living matter. So, pasteurization of food, developed vaccines for rabies, diphtheria and anthrax, and opposed Darwinism, that is the origin of life. So, these are the key discoveries of the Louis Pasteur. Next, John Tyndall. So, John Tyndall developed the technique nothing but sterilization. What is meant by sterilization? Sterilization is nothing but killing of microorganisms. So, the sterilization process John Tyndall developed. This process is called as Tyndallization, where repeated heating of the solutions. So, repeated heating kills the microorganisms, but here he observed that deterioration of the nutrient materials takes place in the solutions. And not only this, he observed what is that the solutions, the shelf life is increased compared with the pasteurization. So, he proposed this Tyndallization process, nothing but sterilization technique to prevent the microorganisms. And he proved that the dust particles carries the germs. So, when the scientists, they are performing this experimental work related to spontaneous generation theory, during this period, this John Tyndall also studied about the spontaneous generation theory, and he proved that the dust carries the germs. Those germs produces the life in the broth material. So, he also contributed to the spontaneous generation theory. Next, Paul Ehrlich. So, Paul Ehrlich, he is a German physician. So, he contributed to the various fields. What are those fields? One is the hematology. Hematology is nothing but study of blood, and immunology, study of immune responses, and chemotherapy, development of the drug molecules for treating the diseases. So, if you see here, hematology, he studied about the blood cells and blood cell disorders. So, where he developed these staining techniques. Apart from this Gram staining, so Gram staining was proposed by Christian Gram. So, another staining, acid phos staining and other staining dyes were used for identifying the various types of blood cells. So, he studied about all these blood cells and diagnosed the various this blood related disorders. So, he contributed a lot to the hematology field, and immunology studied about vaccination and immunization process, and chemotherapy, he discovered the drug molecule nothing but salvarsan or arsphenamide. So, this drug molecule is used for treating the disease, that is the syphilis. So, he discovered that this drug molecule is curing this syphilis disease. So, the chemotherapy development, the era of this chemotherapy was started after the discovery of this arsphenamide drug for treating the syphilis. So, Paul Ehrlich contributed a lot to the chemotherapy. So, nowadays also for cancer treatment, we are using several drug molecules for curing the cancer. Next, the discovery related to the soil or microbial ecology, where these two scientists, Vinogradsky and Beijerinck, these two scientists contributed a lot about the microbial ecology. What is microbial ecology? The relationship between microorganisms and the soil environment. So, they discovered this Nitrogen fixing bacteria. Not only nitrogen fixing bacteria, the bacteria oxidizes the iron, sulfur and ammonia. So iron, sulfur and ammonia. So whatever the bacteria oxidizing these particular elements, those microorganisms were observed by Vinogradsky. So similarly, the Beijerinck, he discovered these organisms. He isolated these nitrogen fixing organisms. What are those? Azotobacter and Rhizobium. So these two scientists contributed to the soil microbiology field. Next, the great scientist Alexander Fleming. So because of the discovery or invention of this Alexander Fleming, the antibiotic era was started. Actually, this is the accidental discovery. It is not a known discovery or known invention. This is the accidental invention. So how he discovered this penicillin means, actually he is working on microorganisms. So during World War time, several of the soldiers were affected by infections and wounds. So he is studying what are the diseases affected in the soldiers and what are the disease causative agents. So he is working on particularly the organism that is Staphylococcus aureus. So he grown this organism in the laboratory and he grown in Petri plates and he left the Petri plates in the laboratory and he went for vacation. So after 2 weeks, he came back and he observed that in one of the plate, one contaminant was grown. So here, if you observe, so this is the plate, he grown organism, this is the Staphylococcus aureus, where he observed the contaminant. And what he observed? He observed that first one contaminant was grown. And the second observation, this contaminant is not allowing this particular surrounding organism, not allowing the organism to grow. That is the second observation. And the third observation, this contaminant is producing some chemical. So this chemical inhibited the growth of this surrounding organism and the transparent zone. Here in this picture you can observe the transparent zone was formed. So that is the observation. The contaminant is producing, particularly that is the mold. So this mold is producing the chemical and it formed the transparent zone. That means it is not allowing the growth of this Staphylococcus aureus. So these are the observations observed by Alexander Fleming. So this is the accidental discovery. So he discovered this penicillin antibiotic. So this penicillin antibiotic inhibits the growth of the bacterial organism. So the penicillin is produced by one organism and this penicillin inhibits the another organism. So this is the secondary metabolite. It kills the other organism. So the antibiotic era was started after the discovery of this great scientist Alexander Fleming. Next is Salman Waxman. So after Alexander Fleming, another antibiotics, several antibiotics. So after Alexander Fleming, several scientists, they started working on the antibiotics. So Salman Waxman, he discovered this drug streptomycin. This is the another streptomycin antibiotic produced by Streptomyces griseus. That is the organism produces this antibiotic. And he published the book. It's related to soil microbiology, soil organisms and soil microbiology. He published in the form of book and he discovered various antibiotics. Actinomycin in 1940, streptomycin in 1944 and neomycin in 1949. All these are the first generation of antibiotics. So now we are using the latest generation, that is fourth generation of antibiotics. So penicillin is the first antibiotic discovered by Alexander Fleming. Later several scientists, they discovered various antibiotics. And these are the various events in the development of microbiology. So far we have seen the scientists and their inventions and discoveries related to the microbiology and its allied field. So historical timeline, we will see the overview of the developments in the microbiology field. So if you see, Antony von Leeuwenhoek discovered microorganisms in the year 1676 and he coined the term animalcules. So in 1857, so Louis Pasteur, he studied about the fermentation process, that is particularly lactic acid fermentation. And another one, the microorganisms, they are not arised by spontaneously. So he disproved spontaneous generation theory and proposed biogenesis theory. And Joseph Lister, he developed antiseptic surgery. And Miescher, he discovered nucleic acids, the genetic material DNA and RNA. And Robert Koch studied about germ theory of disease, where he studied about anthrax and causative agent, that is Bacillus anthracis. And another scientist, Lavron, discovered Plasmodium, the causative agent of malaria. And Koch developed several techniques for culturing the bacteria on the solid surface using gelatin as well as agar. These are the various discoveries in the 19th century. And next, Koch developed the postulates for identifying the relationship between disease and disease causative agent. And Metchnikoff, he developed this phagocytosis. He studied phagocytosis. What is phagocytosis? The macrophages engulfs the bacteria. So this Metchnikoff, he described this phagocytosis process. Gram stain was developed by Christian Gram, and this Petri dish device was developed by Robert Koch student. He is Richard Petri. And Bazarink and Vinogradsky, they isolated nitrogen fixing bacteria, nothing but root nodule bacteria. They are Azobacter, Rhizobium and Nitrobacter, Nitrosomonas. Like this, they isolated several bacteria play a role in the soil microbiology. And he also proved that virus particle causes the tobacco mosaic disease in the tobacco plant. Fleming discovered lysozyme, not only the antibiotic penicillin, he discovered the enzyme, that is lysozyme. So this lysozyme and the penicillin both are having similar mechanism of action on the bacterial cell. They break down the peptidoglycan structure, the cell wall material. So the bacterial cell wall is made up of peptidoglycan structure. This lysozyme and penicillin breaks the cell wall. And first edition of Berge's manual was released, and Griffith discovered bacterial transformation. Fleming discovered antibiotic penicillin, and Ruska, he developed this TEM. What is TEM? Transmission electron microscopy. So because of this electron microscopy, the virus particles were the viruses were observed using this electron microscopy. The Stanley crystallized this tobacco mosaic virus because of the invention of this microscopy. So the scientists, they observed the virus and viral characteristic features, and they studied about the viruses. And next, Avray, he showed that the DNA carries the information, and this play a role in the transformation process. And Waxman discovered the antibiotic, that is streptomycin. And Watson and Crick. So the double helical structure of the DNA was proposed by Watson and Crick, and Cohen et al. They use these plasmid vectors for cloning the genes. And development of this scanning tunnel microscopy, SEM was developed, and the polymerase chain reaction. So this is developed by Kary Mullis. So this polymerase chain reaction is nothing but amplifying the genetic material DNA. So this polymerase chain reaction is used in our molecular biology techniques, not only in this forensic laboratory, also for identifying the suspected DNA material. This polymerase chain reaction is used. And the first human gene therapy testing was begun in 1990, and the discovery of a large bacterium, that is Thiomargarita nambiensis, the genome was discovered after the E. coli, and the discovery of Vibrio cholera has 2 separate chromosomes was identified in the year 2000. And now the summary of this historical events, inventions, discoveries in the microbiology eras, particularly the discovery era and the transition era and the modern biology. So, if you see, Antony von Leeuwenhoek identified or discovered the living organisms. So, because of this discovery only, the microbiology came into existence. So, he identified single-celled organisms, animalcules. Robert Hooke coined the term cell. Edward Jenner proposed vaccination, and Joseph Lister proposed this antiseptic surgery. Robert Koch studied about the diseases tuberculosis, cholera and anthrax, and he proposed germ theory of disease and postulates for identifying the disease and disease causative agent. And Louis Pasteur studied about fermentation, and he developed fermentation process, vaccination and pasteurization process. And Paul Ehrlich, he studied or contributed related to the hematology field, immunology and chemotherapy. And Vinogradsky and Bezering, these 2 scientists discovered this soil organisms, nitrogen fixing organisms, and these nitrogen fixing organisms play a role in the fixation of the atmospheric nitrogen. And finally, Alexander Fleming discovered penicillin. So because of this discovery, the antibiotic era was started. So these are the historical events and contributions by various scientists in the microbiology field. And now the study questions. Enlist the discoveries related to microorganisms and microbiology field, and describe the inventions of Edward Jenner, vaccination process development, and what are the discoveries of Robert Koch related to practical bacteriology, and how Louis Pasteur contributed to the microbiology field and describe his inventions, and how the penicillin was discovered by the great scientist Alexander Fleming. Go through all these questions, refer textbooks and prepare for exams. So these are the various references collected for developing this digital content. So we are acknowledging this public resources and public domain for developing this digital lecture. So thank you all.